Today I'd like to give an introduction to GBM research. So to start, we'll first discuss the basics of GBM. GBM stands for glioblastoma multiforme, otherwise known as glioblastoma or glioma. It is the most common adult malignant brain tumor. That is an important distinction to make because there are other brain tumors that are much more common than GBM. However, if you talk about adult primary malignant brain tumors, GBM is by far the most common. It comprises approximately 52% of all brain tumors, which means that if we had 10 adults that presented with primary brain tumors, about 5 of them would likely have GBM. It is considered a grade 4 brain tumor on the WHO scale. This is a scale of aggressiveness, so the further down the scale we go, the more aggressive our tumor gets. A grade 1 tumor is relatively low proliferating, relatively non-invasive. However, as we move down to grade 4, which is what GBM is, we have a proliferative, highly invasive tumor that also causes necrosis in the brain tissue and leads to vascular proliferation within the brain tissue. Together, being as common as it is and as aggressive as it is definitely something that we want to study further to help our patients. So GBM comes from astrocytes, therefore it's called an astrocytoma. So this here is an astrocyte. It's one of the support cells of the brain, and it usually develops de novo from normal cells. However, there are some cases, just to highlight, that can derive from lower grade tumors known as oligodendrocytomas. These are lower grade tumors that come from oligodendrocytes in the brain and then eventually convert to become GBM. So with that established, let's talk a little bit about how we treat GBM. So typically patients come in and they get a diagnosis based on an MRI. The MRI will show an enhancing region that is a tumor, and they will then be sent for surgical resection. So during this, they try to remove as much tumor as they can. And then they follow this up with TMZ, which is chemotherapy and radiation. And this is meant to together get rid of most of the cells of the tumor. And patients usually do pretty well with this. So usually the tumor will disappear almost entirely, the symptoms will go away, and for an average of 21 months, patients are doing really good. But then, after 21 months, almost 100% of patients have a recurrence where the tumor comes back, the symptoms come back. Some patients go through this multiple times. Some patients go through it just once. It's hard to know. But this recurrence, or the multiple recurrences, are eventually fatal for almost all patients. So the current median survival for GBM patients is about 21 months. And that's something that we really hope to improve with our research. So let's talk a little bit about this treatment. Why is it that the tumor always comes back after therapy? Why is our therapy not effective? And what can we do to make a better therapy for our patients? So the one biggest barrier that everybody talks about with any sort of brain disease, this applies to all brain diseases, not just brain tumors, is the blood-brain barrier. So if we think about a normal blood vessel is just a circle of tissue. The blood is inside. Any drugs or nutrients that it's carrying are easily able to diffuse in and out of the cell and easily able to go in and out of the organs that they're targeted at. However, when we talk about the blood vessels in the brain, they are a lot more protected. So they have a lot more cells around them, a lot more junctions around them. And the point of this is to prevent harmful molecules from going in and out of the brain because you really want to protect your brain. Unfortunately, this also means that some of the drugs that might be helpful for treating tumors like GBM can't really diffuse in and out of the brain as easily as we would like. So it means that a lot of our therapies are actually completely ineffective just because we're going after a tumor in the brain. So this is one thing to be aware of with any therapy that we develop. Another really important thing in GBM research is the concept of heterogeneity. So heterogeneity is considered pathognomic to GBM. Every single GBM tumor is extremely heterogeneous. So what does that mean? So let's go to this diagram here. If this is our primary tumor, we can see that it is composed of multiple different groups of cells. So here we have this orange population, then we have some blue cells, we have some green cells. These are all different types of cells that are composing our primary tumor. And when we give TMZ to this tumor, unfortunately, although some of the cells will be very sensitive to TMZ, so we can see that we sort of lose our orange cells, lose our green cells, these blue cells don't seem sensitive at all. So the blue cells still proliferate, even though we've given TMZ, which should have killed off all of our different cells. And so the blue cells proliferate over time, and eventually we get another giant tumor composed of the blue cells because they're not sensitive to TMZ and they're not going to die off. On the other hand, the other thing to keep in mind is that when we give TMZ or these aggressive drugs, they may induce mutations. And so here our purple cell is a mutated cell. And this purple cell, because it's mutated, is not sensitive to TMZ either, and so it is also going to proliferate over time, and eventually it creates a whole new tumor. That is our recurrent tumor. So chemo can induce mutations, and that can also cause recurrence. I really want to focus on heterogeneity because it is one of the biggest things that prevents us from developing a really good therapy for GBM. Having all these different cell populations in the tumor means that it's very hard for us to develop one therapy that can kill off all the cells, because all the cells have these different characteristics and have different sensitivities to different drugs. So heterogeneity is something you always want to think about when you're thinking about developing a new drug for GBM. So how do we know that GBM is heterogeneous? Well, here we have a microarray done by a group that published in Cell, and down the side you have all of your different genes. So these are all your genes. And across the top you have all of the different patient samples. So all of this is patient samples. And when we put all of these patient samples and look at the gene expression, you can see that the gene expression sort of clusters into patterns. So although every single tumor has a slightly different pattern, if you look down the columns, every single column is slightly different, there are these overall clusters that we see in gene expression. And these clusters have been defined as subtypes. So we talk about 3 different subtypes. And these are classical, proneural, and mesenchymal. And these 3 subtypes are sort of how we define thinking about heterogeneity. So we know that at least there are 3 different subtypes, and within those subtypes there's a lot of variation in different tumors. All right, so where does this variation come from? So there are 2 big theories about where the variation comes from. One of them is clonal evolution. So this is sort of a very Darwinian concept of basically survival of the fittest. So what it says is that there are multiple different clones within the tumor, like we just talked about, and some of those clones survive therapy and some don't. And the clones that do survive therapy are able to sort of recapitulate your entire tumor. So let's say you have 3 different clones here, maybe 2 of them get killed off by therapy, but one of them is able to expand and form this whole new tumor once again. And so this is your recurrent tumor. So that's one thought on where the heterogeneity comes from and where the recurrence comes from. The second thought on where this comes from is the cancer stem cell hypothesis. So this is the idea that you have a primary tumor that's composed of lots of differentiated cells and a few stem cells. These purple cells are stem cells. And when we give the radiation and the chemotherapy and the TMZ together, these lead to cell death for most of the cells, as we've talked about. However, some of the cells survive and actually become more stem-like, and the existing stem cells are still there. And so together, these cells are able to proliferate and form a new tumor that is the recurrent tumor. And so in the glioma stem cell hypothesis, it's a few stem cells that are able to drive this resistance and the heterogeneity and create a whole new tumor. All right, so knowing that we have a highly heterogeneous tumor and that there are sort of 2 different hypotheses of how we get this heterogeneity. So it's either clonal evolution or it's the glioma stem cell hypothesis. But either way, we end up with a really heterogeneous tumor that resists all the therapies that we have right now. We really want to think about how can we find better ways to treat GBM. So how do we develop new therapies that might do better? So for developing new therapies, there are a lot of different research strategies that go after this. So The question behind all of these research strategies is either to find a better way to treat the primary tumor such that it never comes back, or to better understand why is this tumor coming back after TMZ and what can we do to stop that? So what can we do to prevent this recurrence? Or what can we do to just do a better therapy up front so we never have a recurrence that we need to worry about? So one way of looking at this is thinking about new pathways, right? So trying to find new targets, new pathways that we might go after that might help remove the recurrence, that might help or just treat that tumor better from the beginning. Another thing that we could go after is specifically these clones or the glioma stem cells. So we talked about how these clones or the stem cells are driving therapeutic resistance and they're what are allowing the tumor to come back. So maybe we just need to go directly after these, figure out what it is that drives these and go after these. And if we do that, we might be able to remove the recurrent tumor. Another strategy is to identify biomarkers. So maybe we need to personalize therapies a little bit more. Maybe we shouldn't be giving every single patient TMZ. We should actually be figuring out what kinds of mutations they have and what the genetic structure of their tumor is, and then going specifically after that for each patient individually. Another avenue of thought is that we already maybe have the drugs out there that could work, or maybe we need to mix together a few drugs that we have out there. But we basically need to repurpose drugs that already exist towards fighting this disease. And so there are a lot of different trials out there that are screening existing drugs against glioma patients to see if any of those drugs might have efficacy. Yet another avenue is to do immunotherapy approaches. So this is basically where you teach your immune system to recognize cancer cells and to kill them, because it's the immune system's job to take care of your body. And so if you can teach the immune system that this is a marker of a cell that is a cancer cell, maybe the immune system can go ahead and target those cells and kill them, and that way you get a really targeted therapy that only goes after cancer cells and doesn't kill all of your normal cells as well. And then finally, there are also other targeting platforms using nanoparticles that also try to individually target cancer cells and avoid normal cells. So all of these avenues of research are very active, and there's a lot of different studies going after each of these different ways and other ways that we haven't mentioned here. But it's still an open area of research. We still haven't found anything that really is able to stop that recurrence or prevent or better understand why it's recurring after TMZ. That question. Okay, so for all of this research, I want to go over a few of the models that we use. So there are 3 different main types of models that I'm going to talk about in the next few slides. The first one is cell models. So these are cells that we grow in dishes that are supposed to replicate what the tumor cells look like in the brain and allow us to sort of screen drugs to look at different reactions, different pathways, and get a sense of what is going on. The second model is our animal model. So this is supposed to be the model that's closer to humans that we usually use to test once we have something that we think might work. And then finally, I want to go over a few of the data sets that are available for GBM. There has been a lot of amazing research into glioblastoma, and there are a lot of interesting online resources that summarize patient information, patient data, and allow us to see how different genes are working in these large data sets. So let's start first with talking about cell models. So there are 2 kinds of main cell models that we want to talk about. The first one is commercial lines. So these are lines that you can go and buy from a company. So you might go to Sigma or Millipore or Thermo Fisher, any company, and say, I want to buy these cells. And so they'll send you the cells in vials, and you will basically thaw those vials and plate them out. And then you can culture these cells basically forever. The big problem with these cells is that eventually they sort of lose the characteristics of GBM. So people say that they are not the perfect model for GBM because they, over time, start to differ from the main tumor. The other set of lines that we can use is patient-derived xenograft lines. So these are PDX lines, and they're usually called GBM and then a number. So GBM 43, GBM 6, GBM 5, GBM anything is a PDX line. And the way PDX lines work is that they come from a patient, and then we take the tumor and we basically put those cells into a dish and culture them. And then from there, we inject the cells into the flank of a mouse. So this provides sort of a biological microenvironment that really allows the cells to grow over time the way they would in in a brain, or at least as close to a brain as we can get. And then we remove the cells from the flank and put them into a dish, and we're able to culture these cells. The problem with these cells is we can only use them for about 3 to 4 passages. Some people say you can use them for about a month or 2, but the point is that there is a significant limit on how often you can passage them. Whereas these cells you could culture forever, these cells have a limit. And then once you reach that limit, they need to go back into the flank. And so we're going to go back into the flank here. We're going to grow the cells up again and then take them out again, and then we can do experiments on them. The point of this is to keep them fresh and to keep them as close to a real tumor sample as possible, because they have to grow as tumors in the flanks of these mice. And so we're getting as close to a real tumor as possible with these cells. So these are a better model for sure, but they're also a little bit harder to work with. So this is just a reference, but it's a great table of all the different GBM lines that are available. The ones that are highlighted are the ones that we use most commonly in our lab, but these are all lines that you can get. This table just summarizes some of the key mutations that we know are important in GBM. So you have P53, P10, PDGFR, EGFR, and so you can kind of see the key characteristics of these cell lines if you ever need to know which cell line has which modifications or which gene expression patterns. They're divided into 3 groups, and so we have our proneural cells right here, and then our classical cells right here, and our mesenchymal cells right here. And as you remember, these are our 3 subtypes, and it's important because the subtypes each have different clusters of gene expression, and so it's helpful to have cell lines from all 3 when you're doing any sort of major experiment. All right, so now let's move on to animal models. So the model I'm going to go over is a simple model of a brain tumor in a mouse. So in this model, which we use very commonly in our lab, we take a PDX line and we prepare the cells in tube, and then we basically inject them into the mouse brain. And we use like a specified amount of cells we inject in a particular place every time, so we know exactly where a tumor is going. We then wait 7 days to allow engraftment of the tumor, and we can check this by doing an experiment called BLI, which allows us to see the tumor engraftment in the mouse before moving forward. Once we see that engraftment, we usually do a 5-day treatment. So this can be TMZ, it can be any sort of experimental treatment, anything that we're interested in at this point. And then we wait somewhere between 20 to 90 days. In our experience, if we inject 100,000 cells, we usually see about 21 days before the controls die, and about 40-ish days before the TMZ mice die. So we wait X amount of days, and we monitor the mice every day. We look for clinical symptoms of the tumor, and once we see that the mice are close to our endpoint, they have large tumors, we take them and look at the tumor and see for whatever readout that we were interested in with this experiment. So how do we know this model is good, right? Because we have to know that this model actually mimics what's happening in patients, so that we know that it's a good model for us to study everything else we're interested in. So the way we tested that is by doing 2 in vivos, GBM43 and GBM6. So these are both PDX lines that we've talked about. This one is a proneural line, and this one is a classical line, but they're both PDX lines. And so they're supposed to mimic very closely actual patient cell lines. And here we've treated the mice with DMSO, which is our control, and then TMZ, which is our experimental treatment in this case. And if this mimics what happens in patients, then we should see that with TMZ, the tumor sort of goes away as it does in patients, and it should come back after a certain amount of time. And that recurrence should be what eventually causes the mice to die. And that is in fact what we see here, because our DMSO control mice, they get a primary tumor, and then that primary tumor is fatal for them. And so we're seeing that loss of survival here. But if we give them TMZ, then that primary tumor actually goes away, which we can follow with BLI and see that the tumor actually does go away. And then here we have a recurrence, and then the mice die a little bit later, which is the same pattern that we see in our patients. So that tells us that this is a good model that we can trust and use to test other therapies. All right, and then I want to go over a few different options for online data sets. So as I said, there has been a lot of really good research into GBM. And from that research, people have been able to take de-identified patient genetic information and put it into these data sets that we can use to understand how different genes work in GBM and how different patients, different elevations in genes or different depressions in genes might result in different phenotypes. So for GBM, there is this great interface called GlioVis. And what this is data from about 500 patients organized into different data sets where we can look at various different options. So here we can look at expression and we can look at survival. So this is sort of your interface when you go to the GlioVis website, and you start by clicking Explore to look at the different genes. We're going to look at adult genes because we're interested in GBM. You can choose your data set here. And then you can choose what kind of tumor you want to look at and what gene you're interested in looking at. Here you have some options for statistics and visualization, which are sort of secondary to understanding how this data set actually works. So let's talk about the different options that you have. So first, it's important to understand that GlioVis looks specifically at RNA levels. So when we talk about biology, we know that we have DNA, which is the main genetic code. That becomes RNA, and then that is translated to protein, which actually makes changes in the cell. And GlioVis, for our purposes, for what we're going to talk about right now, is looking mostly at this RNA level change. Okay? There are 3 data sets that I think are particularly good. So the ones that I like to use are CGGA, TCGA-GBM, and the TCGA-GBM-LGG. And we're going to go over each of these in greater detail. But first, I just want to highlight what an amazing project this is. So TCGA, it was quite an incredible project that looked at 33 different tumor types across 11,000 patients and did 7 different types of analyses, which are listed here, in order to really get at what is changing in these tumors and what genes are expressed, what genes are not expressed, how can we better understand what's going on with these tumors. And it provided a wealth of data that researchers use still today for all these different cancers to try to understand what's going on. We are specifically interested in GBM, and for GBM there are about 600 patients in this data set, and we can look at all of these different things, but what I'm going to focus on today is really the mRNA expression and the RNA-seq part of this. So let's start by talking a little bit about the 3 data sets. So like I said, there's CGGA, there's TCGA-GBM, which means it's exclusively the GBM patients, and there's TCGA-GBM-LGG, which means it's both high-grade and low-grade glioma patients. So this includes low-grade tumors as well. So CGGA has many different tumor types. It's useful for sort of looking at gene expression across multiple types of tumors, and it allows you to really break down whether you're looking at primary versus recurrent tumors, which is a big plus. The TCGA-GBM dataset looks at patient data specifically in GBM, and it offers multiple platforms. I usually use RNA-seq, but all of them are fine. And then the TCGA-GBM-LGG combines the high and low-grade gliomas. If you're interested in looking at higher gene functions across all types of gliomas, it's an interesting data set to look at. For your tumor type options, which is your first option along the side column, you can choose for the Chinese Glioma Atlas whether you want to look at primary or recurrent, or secondary, which is usually only GBM patients, or you can look at all of these together and just select all patients. And then for all of them, you have different options for your plot type. So you can choose to look at it by histology, by grade, by recurrence, or by subtype, or by copy number for the TCGA ones. So this allows you a lot of freedom in how to organize your data and what kinds of visualizations to generate. So let's look at some examples of this. So let's start again by going over the different types. So when it asks you what tumor type, what you should think of is basically asking you which patients to include. So if this box is all of your patients, these are all the patients that you could choose from. Within that, you have a set of primary patients who have just been diagnosed with their first tumor. You have a set of patients who've only received one therapy round. So these are patients that are coming back with secondary tumors. And you have patients that have had a recurrence in general. So these are patients across all tumors who've had a recurrence of their tumor. So you can choose any of those individual populations, or you can choose to look at all of the patients. And there are different scenarios where this is useful. So for example, if you were interested in a gene that you know is involved in driving primary glioma, it's a gene that's really important when the tumor first establishes itself, then you might want to look at primary. But on the other hand, if you're looking at a gene that you know only comes up after they've seen TMZ or after the patient has gotten some sort of therapy, then you might be interested in looking specifically at recurrence. On the other hand, if you just want to know how this gene does across the board or you're not quite sure yet, you might want to start with looking at all patients. You should think of plot type as how to organize your plot. So this is asking, okay, like these are the patients that you know you want to look at. I know that you want to look at all the patients, or I know you want to look at only recurrent patients, but how should I organize those patients? So it gives you the option of organizing by histology, which means sort of by tumor type. It gives you the option of organizing by grade, so that's going to be the WHO grades we talked about. It gives you the option of organizing by recurrence, then you can look at primary versus recurrent. And finally, gives you the option of organizing by subtype, which would be the proneural, classical, and mesenchymal that we talked about. All right, so now that we've sort of gone over your different options, let's talk about the graphs. So here I've plotted all of the graphs for P53. The importance of P53, although I'm sure most of you know this, is that if P53 is mutated, that means that it's not performing its normal function. So normally P53 acts as a tumor suppressor, but a lot of tumors will mutate P53 in order to make themselves more aggressive, because then P53 cannot turn off their growth anymore. So P53 mutations are associated with generally more aggressive tumors. So here our first plot is we chose any type. So here I chose all patients plus the histology. So what this means is it breaks down the tumor by histology and gives me expression of P53 for every single different histology. So this is my mRNA expression along the y-axis. This is how strongly the gene is being expressed. And these are all of my different types of tumors here. And what we can see is that GBM, for example, shows pretty high P53 expression if we compare it to this oligoastrocytoma here. And so this is useful for seeing how your gene does across different types of tumors. Another thing we can look at is grade. So if we want to know if P53 goes up with aggressiveness, like I told you it does, maybe we want to look at grade because this is increasing aggressiveness. And we can see here that the expression is progressively going up with increasing aggressiveness. We can also look at recurrence. And so again, this would be all patients. And we are going to look at how the gene does with more recurrent tumors. And so we can see that as the tumor recurs again and again, we're seeing an increase in P53 over time. And finally, we can look at the subtype. Sometimes your gene might be a gene that is only expressed in a particular subtype or is only important in a particular subtype. Here, I don't see huge differences. The proneural is perhaps a little bit down in expression compared to the other two, but there are cases where you know that a certain gene is very important for a particular subtype, or you might want to make sure that you're doing your experiments in the correct cell line to capture the effect, so you'd want to check if there's a specific subtype that would be good for your gene. Okay, so now let's talk about survival. So we just talked about expression. The other big thing that you can see in GlioVis is survival. And there are two different cutoffs for survival. So when we look at GlioVis survival data, you can either have a median cutoff, which is the standard, or you can have an optimal cutoff. The median cutoff just cuts at the middle. So whatever the middle is of the RNA expression, that'll just cut right in the middle of that. And then it will give you, these are all your patients that are high expression, and these are all your patients that are low expression. And that low expression gets plotted as the blue curve, and the high expression gets plotted as the red curve, and it just shows you survival. So how long did all of the patients who were high, how long did they survive, versus how long did the patients that were low, how long did they survive? And so what we see here is that if P53 is low, the patients survive longer. which is what we might expect if P53 actually does track with aggressiveness. When you do optimal cutoff, you do see that the program basically tries to give you the best separation of the curves. So it moves the cutoff and figures out where you're going to get the most significant separation of curves, and that's what it plots. So here it set the cut point lower than it did at the median cutoff, and we can see that we have this many patients for a low and all of these patients for the high. And in this case, it's actually showing the opposite result. It's showing that if P53 is elevated, patients survive longer. And I put this example on here to really highlight how important it is to be careful with interpretation of these curves, because this first one kind of tracks along what I've said. So this one, if we look at it, it matches what we think we should see. But this one, our optimal cutoff, is the complete opposite. So one important thing with this is to keep in mind that no one graph is perfect. And so if you really want to make a statement about survival, you should have at least 3 to 4 graphs that show the effect that you're interested in. If all 3 of them show it across 3 different data sets, chances are you're onto something. The other thing is to be careful with the median versus the optimal cutoff. So some people prefer median because it's sort of unbiased, and so, every time if you just cut exactly in the middle and you take whatever that result gives you, you're not really biasing yourself in any way. However, other people prefer optimal cutoff because you want to give yourself the best chance possible. You want to see if there's any effect at all with this gene. And so if you're going to be looking at genes anyway, you might as well look at the best option or the best outcome for your gene and go based on that. So it's very much a subject of debate. People think both are appropriate to use, but you have to be aware that there are multiple different pros and cons to each one, and that you want to interpret it in the context of at least a few graphs so you can be sure of the effect that you're seeing. Okay, now let's talk a little bit about another great resource called Protein Atlas. So as I talked to you about before, the central tenet of biology is this DNA to RNA to protein. And we said that GlioVis is measuring RNA. So RNA is very helpful because it shows us what gene is being expressed, but protein is the best thing to look at, right? Because this is what is actually going to do something in the cell. And it would be nice to have a way to look at the protein. So Protein Atlas allows for that. And it's a very, very detailed atlas, has lots of great data, is a really, really great resource. And if we go to the Protein Atlas website, this is kind of what you'll see when you start. So you can go ahead and enter your gene. So let's say we entered P53 here, or LNX1, or whatever gene was of interest, and clicked enter. Then we would get this new interface. And so this allows us to see tissue. So this would be our normal tissue if we clicked on this. This will allow us to see how the protein is expressed in cells, so it's really useful for localization. And then this will allow us to see different cancers. It's not just GBM, it's all kinds of different cancers. So let's start by talking about the pathology section and the normal section. So when you click on pathology, if I clicked on pathology in the previous slide, I would have gotten this page that shows me all of the different cancers. So here you have all your different cancers. And on the side here, you have your expression level. So this, as a graph, is a really useful tool for seeing if your gene is elevated in a particular cancer. For example, here, this one is elevated in thyroid cancer for sure, but it's kind of about the same in all of our other cancers. If I'm interested in looking at glioma specifically, I can go ahead and click on this glioma button, or the glioma graph, and then it takes me to a page that actually has these really nice sections. So these are patient sections that have been stained with the antibody of interest. So if I had put in LNX on the previous slide and gone and clicked on glioma, and now I would be seeing that these are LNX1 stainings in glioma. So I can see that these are really brown, which means that I have really high expression of my gene of interest in glioma, which is a very useful thing to know about a gene that I might be studying. You also might want to compare these to normal, right? Because if your gene is just expressed highly in glioma, but also expressed highly in normal, then it's not really changing in GBM. So you want to always go and compare this to normal expression. So for normal, if we clicked on the blue part of the previous slide, which is our normal, then we would get this diagram. So this diagram shows you sort of a human figure, and then it shows you all of the different tissues available for you to look at. And so we're really interested in the brain, so we're going to look at the cerebral cortex for our normal. And again, we'll get taken to this page that has these sections, and these are our normal sections. And here for our gene of interest, we can see that it's pink, so it's really low expression. And so we know that in GBM compared to normal, we're seeing a really high expression of our gene in GBM and really low expression in the normal tissue. So you can see how this would be a really useful tool for looking at patient gene expression in multiple genes that you're interested in without having to do a lot of work by yourself. So if I was interested in a particular gene, I wanted to make a case for it, I could pull all of the information from GlioVis and show that, for example, it's elevated in GBM, show that it's elevated in GBM at the protein level with this protein atlas data, show that there's a survival benefit with the gene using the GlioVis data, and together that makes a really nice case for why I might want to go after a particular gene. And then this here is just another example just to show you that in IHC Brown, the more brown your tissue is, the more positive it is. And so this is a highly positive tissue for whatever antibody it was stained with. And this is sort of the negative normal tissue that you can see is a lot more pink or purple. This is just for your reference to help you understand. All right, and then to wrap up this lecture, I just want to talk a little bit very quickly about good experimental design. So, the core of research is that you have some sort of question or hypothesis, and you want to go and test it with your experiment. So you want to figure out how to design a really good experiment that actually answers the question that you're interested in answering. So these are the things that I think are most important to think about when you're actually designing an experiment. So the first thing you should always know is, why am I doing this experiment? So you should have a clear question that clearly states what it is that you're trying to find out and what you think you're going to find out from this experiment. The next thing you want to know is what model you're going to be using. So are you going to go with cells? You're going to go with animals? And if cells, like which cells, which animals, exactly how are you going to do this? That's another really important thing. The next thing you want to think about is the course. So how much of a specific drug are you going to use? How long is your experiment going to last? How are you going to maintain it? And most importantly, what are the controls going to be? Any good experiment has positive and negative controls. Without a good positive control and a good negative control, it is really hard to know if your experiment worked, and it's hard to draw any conclusions. And so if you learn nothing else, the most important thing about designing a good experiment is that it has really, really solid controls. The other things to think about would be how you're going to read out and analyze the results. So what does the analysis look like, and how do you make sure that it worked? And then what do you think this experiment is going to give you? You should never do an experiment unless you have a clear-cut question that you think it's going to answer, and you have a clear-cut way in which it's going to help your project. So this here is just a quick example of how you might design an experiment. So you will see that we've said that we're interested in how GBM43 responds to TMZ. And the why for this would be that GBM43 is a really common cell line that we use for glioma research. TMZ is the standard of care drug, so it's the drug that all patients get. And so it's really useful to understand how GBM43 cells respond to TMZ in culture, because it's going to be the base of a lot of our experiments. And so here we've clearly specified how we're going to grow the cells, what kinds of cells we're going to use, and how long we're going to grow them for. We've talked about the different doses that we're going to use of TMZ. And again, we have our replicates. We make sure to specify our controls, because that's really important, and specify the timing. We talk about our method of analysis. And we know that we expect that as the TMZ goes up, we'll see increased cell death for GBM43. So that's our expected result. So just to review, today we've talked a little bit about GBM as a disease and about the treatment options available. We've gone over at a very high level the different approaches that people might take to finding a better therapy. We've talked a little about the different models and resources available. And then we very quickly did some basic experimental design, talked about some of the things that you want to make sure to think about before doing an experiment. If you have any questions or feedback, please feel free to contact me, and feel free to follow us on Twitter to see more of this. Thank you so much for listening today.